Hello students, welcome to EPG Pathshala. I am Dr. Manisha Pandey from School of Sciences, IGNOU. I am a biochemistry faculty. Today we are going to talk on module 3, Biosafety and Biocontainment 2 under the paper Molecular Therapeutics. In the first module, we discussed about the various work practices, personal and protective equipments, and biocontainment facilities which are required to ensure biosafety of an individual. In this module, we'll be discussing about the regulatory frameworks. So, after going through this module, you should be able to discuss the regulatory frameworks in India as well as some of the regulatory frameworks abroad which are in vogue for modern biological research and its safe application. You would also be able to explain the various functions of various statutory committees which work to ensure the biosafety and biocontainment and are in place by the virtue of Environment Protection Act 1986 and the rules 1989. You will also be able to describe in detail the composition and function of Institutional Biosafety Committee, which is at the institute level. You can discuss and explain the Cartagena protocol on biosafety. Then we'll also be able to describe the environmental impact due to GMO and LMO, and the negative part of it, the bioterrorism and biological warfare. Since we talked about the 4 components of biosafety in the previous module, the 4 components were administrative control, work practices, personal protective equipments, and engineering control. We discussed the later 3 into the previous module. In this module, we'll talk about the administrative controls, that is, regulatory practices, the various guidelines, rules and regulations, security clearances. So, we will be talking about the Environment Protection Act, which is an umbrella legislation. We will be talking about Rules 1989, which is about the microbial uses recombinant DNA products which are being used for creating the genetically modified crops or animals. So, let's try to define biosafety differently. Till now, we have been defining the term biosafety as application of safety measures to protect ourselves, to protect a researcher who is working on a potentially biological hazard, and to limit the contamination of the work environment as well as community. So, we have talked about biosafety of an individual. When we say about regulations, it can be defined in a different way. What is biosafety? Biosafety is protecting humans and animal health, protecting the biodiversity from the possible adverse effects of the products of modern biotechnology. Because in modern biotechnology, we are using recombinant DNA, we are altering the genome of one individual or organism or plants. So here we will be focusing on another aspect of the biosafety, that is protecting the human or animal or the biodiversity from getting polluted or say genetic pollution. Since we were talking about biosafety, biosafety of individual researcher as well as the biosafety or safety of the environment and community as a whole. And we have also mentioned about the administrative controls which are in vogue to ensure the safety of individuals and environment. So let us look what is the significance of biosafety regulations. How do they work? Biosafety systems actually they ensure that application of biotechnology continues to be safe and it does not expose the employees, community or the environment to any avoidable ill effect. These regulations also describe the policies and procedures which, when adopted, they ensure the safe application of modern biotechnology, which is beneficial for the mankind. Now we look at the biosafety regulatory systems in India. The safety regulations for environment was brought down in the year 1986 by the Act of Parliament. In the year 1986, Environment Protection Act was given by the Ministry of Environment and Forest. This act was like an umbrella legislation under which then many rules and regulations were enacted. For example, 1989 rules, which is about the manufacture, use, export, import of various hazardous microbiologicals. Then came Drugs and Cosmetics Act. Number of acts are there, like Schedule Y of Drugs and Cosmetic Acts, which apply to drugs and pharmaceuticals, Seed Policy, Protection of Plant Varieties and Farmers Act, Food Safety Standards. So many acts and rules came which ensured that biosafety of individual as well as environment is protected. If we look at the international perspective, the International Biosafety Regulatory System, there is a Convention on Biological Diversity. The CBD is an internationally legally binding treaty with three main goals: conservation of biodiversity, sustainable use of biodiversity, and fair and equitable sharing of the benefits arising from the use of genetic resources. This Convention on Biological Diversity is dedicated to promoting sustainable development. Convention also recognizes that biological diversity is about more than plants, animals, and microorganisms or their ecosystems. It is about people and our need for food security, medicines, fresh air and water, shelter, and a clean and healthy environment in which we live. From the Convention on Biological Diversity, there is a Cartagena Protocol, which was the outcome of CBD. Cartagena Protocol is related to biosafety. Another regulatory framework is Codex Alimentarius. Codex Alimentarius is a Greek word which means food safety. So here also they have laid down various standards related to foods which are practiced. So we were talking about the regulatory framework in India and abroad. Let's discuss the regulatory framework or the act which is in place in our country. The Environment Protection Act 1986, it came into vogue after the Bhopal tragedy, and EPA Act 1986 is an umbrella legislation for the protection of environment. After the EPA Act 1986, in the year 1989, rules and procedures for handling GMOs and hazardous organisms were laid down. They are popularly referred as Rule 1989. These laid down the foundation of biosafety regulations in India. These legislations, the EPA Act 1986 as well as Rules 1989, they are laid with a view to protect the environment, nature, health in connection with the applications of gene technology and microorganisms. The Rule 1989, its genesis goes back to EPA Act 1986. From the EPA Act, this rule came out later, and it is known as Rules for the Manufacture, Use, Import, Export and Storage of Hazardous Microorganisms, Genetically Engineered Organisms or cell. This rule classifies microorganisms or genetically engineered products into two categories: animal and human pathogens and plant pests. This rule also provides a scheme for approval of various projects in which these kind of hazardous microorganisms or genetically engineered organisms are involved. It covers almost all the activities, including laboratory experiments to release into the field or transboundary movement. There are detailed guidelines related to administrative mechanisms, and if we fail to follow these rules and procedures, there are punitive provisions as well. We were discussing about the rules 1989. This rule is under the Act, EPA Act 1986, and it has been laid down by the Ministry of Environment and Forest and Climate Change. The Rule 1989 envisages the constitution of 6 statutory committee where they have got different roles to perform. One of the committee, GEAC, that is Genetic Engineering Appraisal Committee, it talks about or it gives clearances, laid down the guidelines and procedures related to import, export, transport, manufacture, process, use and sell, whether it is a hazardous microorganism, genetically engineered organism, genetically engineered substance or cell. We were talking about GEAC and Rule 1989 where we were discussing the use of microbiologicals for the benefit of humankind, laboratory purposes, release into the environment. Rule 1989 also explains various terms. For example, microorganisms has been defined as all the bacteria, viruses, fungi, mycoplasma, cell lines, algae, protozoans, nematodes indicated in the schedule and those that have not been presently known to exist in the country or not have been discovered so far. So, microorganisms which are of exotic nature. The term genetic engineering has been defined as technique by which heritable material which does not usually occur or will not occur naturally in the organism or cell is generated outside the organism or the cell and is inserted into the said organism. It shall also mean the formation of new combinations of genetic material by incorporation of a cell into a host cell where they occur naturally, as well as modification of an organism or in a cell by deletion and removal of parts of the heritable material. Rule 1989 defines the term biotechnology as application of scientific and engineering principles for the processing of materials by biological agents to produce goods and services. Cell hybridization has been defined as formation of live cells with new combinations of genetic material through the fusion of 2 or more cells by means of methods which do not occur naturally. That is, the fusion of genetic material from 2 different species, which is not a natural phenomenon. Gene technology has been defined as the application of the gene technique or genetic engineering, including self-cloning and deletion as well as self-hybridization. There are many terms which have been described in Rules 1989. We have discussed some of them. Now let us look at the statutory committees which have been mentioned in the Rule 1989. It clearly defines 6 competent authorities and the composition of such authorities for handling various aspects of rules. The various statutory committees are RDAC, the Recombinant DNA Advisory Committee. It has an advisory role. It advises what sort of project should be undertaken. Then there are 3 committees which has got approval role. They approve the projects to be undertaken at various institutes, laboratories. They are Institutional Biosafety Committee, IBSC, Review Committee on Genetic Manipulation, RCGM, Genetic Engineering Approval Committee, GEAC. Now it is known as Genetic Engineering Appraisal Committee. Other 2 statutory committees are State Biotechnology Coordination Committee, SBCC, and District Level Committee or DLC. The SBCC and DLC look into the monitoring and enforcement of the rules and guidelines which are laid down by the approval committees. Now let us have a detailed look at with the functions of various statutory committees. Recombinant DNA Committee or RDAC, this works under Department of Biotechnology. It reviews various biotechnological developments and projects. It also recommends safety regulations for research, use and application. The three approval committees, GEAC, RCGM and Institutional Biosafety Committee, which have approval role, work differently. GEAC works under Ministry of Environment and Forests and Climate Change, which was earlier known as Ministry of Environment and Forest only. They are involved in providing environmental clearances to large-scale use of recombinant products, recombinant research policies, release into the environment. They also supervise the implementation of the rules and regulations and have punitive powers. GEC permits the use of GMOs and product thereof for commercial applications. GEAC adopts practices for restriction or prohibition, production, sale, import and use of GMOs both for research and applications also under the Environment Protection Act 1986. GEAC has the authority to authorize agencies or persons to have powers to take punitive actions under the EPA Act. If we look at the main functions of the Review Committee on Genetic Manipulation, the RCGM brings out manuals of guidelines specifying procedures for regulatory process on GMO research, use and applications, including in industry with a view to ensure environmental safety. RCGM also reviews all ongoing recombinant DNA projects involving high-risk category and controlled field experiments. They lay down procedures for restriction or prohibition, production, sale, import, and use of GMOs, both for research and application. RCGM also permits experiments with risk group 3 and above with appropriate containment facilities. They authorize the import of GMOs, transgenes, transgenic seeds for research purposes. If we look at the functions of Institutional Biosafety Committee, IBSC, the Institutional Biosafety Committee function at the institute level. If the organism belong to risk group 1, the role of the institution is to inform the committee. If the risk group 2 microorganisms are involved, then they have to seek approval from the higher committees. For RG3 and others, they go to GEAC. Institutional Biosafety Committee also prepares an emergency site plan. They ensure the adherence of guidelines and regulations, and it is the nodal point for interaction with other committees. If we talk about the two committees involved in monitoring and evaluation, SBCC and DLC, the State Biotechnology Coordination Committee, SBCC have the powers to inspect, investigate and punish if there are any statutory violations. They also look into the post-release monitoring if there is a recombinant DNA involved for GMO or LMO. They are the state nodal agency. District level committee or DLC monitors the safety regulations in installations, also monitors the post-release, and they report to SBCC and GEAC. The Cartagena Protocol on Biosafety is a byproduct of Convention on Biological Diversity. In the year 1992, at United Nations Conference on Environment and Development, the Convention on Biological Diversity was held, and biosafety was one of the issues addressed by the convention. Then came the Cartagena Protocol on Biosafety. It was the first international agreement which entered into force on 11 September 2003. India is also signatory to CBD and Cartagena Protocol. Cartagena Protocol aims to ensure the safe handling, transport, and use of living modified organisms resulting from modern biotechnology that result or repeat resulting from modern biotechnology that may have adverse effects on the conservation and sustainable use of biological diversity, taking into account risks to human health and especially focusing on transboundary movements. The Protocol on Biosafety is an environmental treaty and it is legally binding to all the parties. Genetically modified foods are also within the scope of Cartagena Protocol, but only when they contain LMOs and those LMOs that are capable of transferring or replicating genetic material. Cartagena Protocol defines the LMO as any living organism that possesses a novel combination of genetic material and is obtained through the use of modern biotechnology. Although in everyday usage, LMOs are usually considered to be the same as GMO. Since we were talking about Cartagena Protocol and while discussing about the roles and functions of various statutory bodies, we mentioned the term LMO and GMO. So let us have a detailed look. What is an LMO? The full form of LMO is living modified organism. LMO is any living organism that possesses a novel combination of genetic material obtained through the use of modern biotechnology. Like one organism having the genes from some different organism. Generally, in everyday usage term, LMOs are usually considered to be the same as GMO or genetically modified organisms, but definitions and interpretations of the term vary. Both LMO and GMO are the product of modern biotechnology. It includes recombinant DNA and direct injection of nucleic acid into the cells or organelles or the fusion of cells beyond the taxonomic family. Genetically engineered organisms, genetically modified organisms, transgenic organisms, biotech crops, they all can be broadly termed as GMO. Let us have a look at some of the chronological facts involved or having the application of microbiological hazards or say microbiological products for genetic experiments, recombinant DNA, and then its use for the betterment of society. The first recombinant DNA was made in the year 1972 by American Paul Berg. Later in the year 1973, the first GMO was created by Stanley Cohen and Herbert Boyer. Then came the first transgenic mouse and later the first transgenic plant, obviously abroad, not in India. In the year 1976, in US, the first genetic engineering company, Genentech, was established, and in the year 1978, they started producing genetically engineered humulin or human insulin, which was to be used as insulin for those who are suffering from diabetes. After a gap of let's say 15-20 years, in the year 1994, the first genetically modified food, flavor saver tomatoes, were released into the market for consumption after FDA approval in United States. Today, if we go back 2 years, in 2015, the Accu Advantage, a salmon which has been named as Accu Advantage, became the first genetically modified animal to be approved for use as food. In India, the first transgenic crop to be released was in 2002 when the National Seed Policy came. Since we are concerned with the biosafety of the environment as well as community, and we have looked at the chronological facts how recombinant DNA is getting into our daily lives. Let us critically analyze the two. What are the advantages of genetic modification and what are the disadvantages? The genetic modification is contributing to increased productivity of food, feed and fiber. So it has the capacity to make a nation self-sufficient, self-reliant, where we have got the limitation of the arable land. Then it also ensures that our biodiversity is conserved because we are not getting into deforestation. So we are protecting our biodiversity, we are protecting our sanctuaries. Maybe genetic modification will also help in mitigating challenges which are associated with climate change, and thus they'll help to improve our economic, health and social status. But if we look at the disadvantageous part of genetic modification, why we need to ensure so much of biosafety, why we have to practice the biocontainment while working on these biological hazards in laboratory. There are sufficient evidences regarding the long-term effect of these GMOs on human health and environment also. So they may affect our health as well as the environment. Also, GMOs might adversely affect the non-GMO accidentally by crossbreeding. So, when there is a crossbreeding between a GMO and a non-GMO, it might result into a generating organism which is completely new, which has got new set of genes and characteristics. So, it may lead to genetic pollution. We were talking about the pros and cons or advantages and disadvantages of the GM crops or genetic engineering applied to the food and animals. If we look at the global scenario, the global adoption of GM crops have increased a lot since 2 decades. In the year 1996, the global area under GM crop was only 1.5 million hectare, which has increased now to 181.5 million hectare. It means there has been a tremendous increase in the area under GM crop production. If we look at the crop scenario, soybean is leading. Other crops which are being cultivated are cotton, maize and canola. If we look at the country-wise perspective, USA is leading in GM crop production, followed by Brazil and Argentina. Canada and India are competing for the same position, area-wise for GM crop cultivation. If we look at the Indian perspective vis-a-vis the genetically modified crops, here instead of calling GM crops, we prefer calling insect-resistant varieties. In India, IR cotton is being cultivated nowadays. In 2016, approximately 7.2 million cotton farmers adopted the IR cotton, representing approximately 96% of the estimated crop area under this cultivation. There is a rapid adoption rate for IR cotton because it is a high yielding variety. It is insect resistance also. And if you recall, in the previous module on biosafety and biocontainment 1, we were talking about the various risk groups, the microorganisms which are placed in risk groups and involved in research. We mentioned Bacillus thuringiensis. BT cotton, IR cotton is same, where the genes from bacteria Bacillus thuringiensis have been used to develop an insect resistant variety. 11 states in India are growing IR cotton nowadays. To name some of them, Maharashtra is leading, Gujarat, Andhra Pradesh, Telangana, Haryana, Rajasthan are there. We can also say 2016 was a turning point from Indian perspective. India approved the commercial release of its first biotech food crop, mustard, known as DMH11, or Dhara mustard hybrid 11. This mustard is the first genetically modified food crop developed indigenously by the Center for Genetic Manipulation of Crop Plants, CGMCP of the Delhi University. This biotech mustard is a high yielding variety, so it will lead to the solution, or it will provide solution to the farmers where they are suffering from the low yielding plants. Also, India is facing a huge deficit in edible oil. So annually we are importing edible oils from other countries, including oil extracted from biotech soybean and biotech canola. So if we can import oil from other countries which are biotech in nature, why not use these plants in our country? Also, in the year 2016, approval of field trials for IR chickpea and IR pigeon pea was given. These insect-resistant chickpea and pigeon pea have been developed by ICAR, Indian Institute of Pulses Research. So it's one of the developments or advancements in the field of food security. Then in this year, some important guidelines and documents were also released. To name some of them, Guidelines for the Environmental Risk Assessment of Genetically Engineered Plants 2016 document was released. Then monitoring of confined field trials of regulated genetically engineered GE plants was given. Also, specific biology documents for biotech crops and traits under development in the country like potato, rubber, papaya, etc. were given. So we can say 2016 was a turning point from Indian perspective. Since we are discussing the module on biosafety and biocontainment, talking about bioterrorism and biological warfare also becomes important. What is bioterrorism? Bioterrorism is use of infectious agents or other harmful biologicals or any biological substance as weapons for terrorism. Like whatever good work we were planning in the laboratory for the betterment of society, for the benefit of society, is now being used for some bad purposes with some bad intentions. If we look at some of the examples which we can consider into this bioterrorism, use of any psychotic substances to contaminate food is bioterrorism. Use of toxins, poisons for political assassination or killing someone. Raids with crude biological cloud bombs, like using some microbes, real hazards to infect the society. Using dried viral preparation spray powders. Then another aspect of bioterrorism can be disrupting the agriculture, decimating the livestock or contaminating the environment. If we disrupt the agriculture or decimate the livestock, we are actually seeding food insecurity in the society by intentional food poisoning and food infection. What is the attraction for biological warfare? Why biological warfare are considered to be better than chemical warfare, and it is used for terrorist attacks? Reason being, the biological warfare have very low production cost. They are not detected by routine security systems. Then the biological warfares can be easily transported from one location to another. So it is one of the features or attractive features. Then these biological weapons are invisible, virtually weightless. So it helps in concealing it. It helps from getting it detected. Then there is very high cost efficiency index attached to it. A small amount of biological hazard can pose great threat to the society. So it has got high casualty numbers attached to it as compared to the manufacture of a chemical and nuclear weapon. These are the key attributes which make biological warfare weapon or weapon of choice in the hands of terrorists. So students, let us now summarize what we have learned in this module. We have learned about the various regulatory frameworks in India and abroad. So there are two main rules or acts which are in vogue: EPA Act 1986 and Rules 1989, which is about the use of microbiologicals for various purposes. Then we have looked into the aspects of various committees, their composition, their function, whose approvals are needed when we have to work on the microbiologicals. Also, we have discussed the international framework, the international regulatory framework like Cartagena Protocol, and the environmental impact of GMO, how it can increase the food production, how it can solve the food security problem, and also we have talked about its adverse effect, how it is adding to the genetic pollution or it may add to the genetic pollution. And we have also talked about bioterrorism and biological warfare, which is like negative use of the microbiological or microbial hazards. Thank you.